Hey everyone, and welcome to RUBIN'S Mind Lab, where we break down the latest research into what it really means. Ever wonder if we can trust all the studies we read, especially when they're a bit different from each other? Today we're diving into a fascinating idea called Bayesian Evidence Synthesis, or Bayes. Think of it as a super smart way to combine findings from studies that don't quite match up perfectly. It is like trying to put together a puzzle where some pieces come from different boxes, but they're all supposed to fit the same picture. So, what is the big problem Bayes is trying to solve? Well, in science, we often want to combine the results from multiple studies to get a clearer picture. Usually we do this with something called meta-analysis, where we pool data or average effect sizes. But this only works if the studies are really similar. What happens when they're not? Imagine one study measures a treatment success by looking at patient questionnaires, another by looking at brain scans, and a third by observing behavior. These are all valuable, but how do you combine them if they're so different? That is where BS comes in. It is a way to combine evidence from studies that can't be pooled easily. The key idea is that each study, even if it looks at a part of a bigger picture, is still testing the same core idea. BS uses something called Bayes factors to measure how much evidence each study provides for a specific hypothesis. Then, it cleverly combines these evidence scores from different studies. Instead of just saying, is there an effect or not, Bayes lets us test more specific predictions. For example, instead of just testing if a new therapy is better than a placebo, we can test if therapy A is better than therapy B, which is better than therapy B, which is better than therapy C. These are called informative hypotheses, and they're like building blocks for bigger theories. The researchers in this paper wanted to know if BS really works when studies only look at parts of these big informative hypotheses. Think of our puzzle analogy again. What if one study only tests if piece A is bigger than piece B, another tests if piece B is bigger than piece C, and you need to know if A is bigger than B, which is bigger than C, and so on. Can Bayes reliably tell us the answer to the whole thing? To find out, they ran a big simulation study. Imagine they created a true scenario like a perfect ranking of 4 things. Let's call them mu, 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 and mu. Then they pretended that different studies only tested parts of this ranking. For example, one study might only test if mu is greater than mu, another if mu is greater than mu, and the third if mu is greater than mu. Together, these 3 partial tests cover the whole ranking. They then compared the results from BS, which combined the evidence from these partial studies, to a traditional approach where you'd ideally have all the data together to test the complete hypothesis. They did this in 2 main ways. First, they used the exact same data for both BS and the complete hypothesis test to see how similar their conclusions were. This is scenario A. Second, they simulated entirely separate studies for each partial hypothesis and then compared that to a separate complete hypothesis test, which is more like a real Zen world situation. This is scenario B. They also tested 2 main conditions. Condition 1, where the true ranking was the same as the one they were trying to test. This is like checking if your puzzle pieces actually build the picture you expected. Condition 2, where the true ranking was different from the one they were testing. This is like realizing your puzzle picture is actually different from what you thought. Let's look at the results. In condition 1, where the hypothesis was correctly specified, things looked pretty good when they used the same data for both methods. Scenario A, the differences between BS and the complete hypothesis test were generally small. They got even smaller as they increased the sample size or the effect size, which is like how strong the true differences were. In scenario B, where they used separate simulated studies, the results were even more encouraging when the hypothesis was correct. Best results, PMPBs, closely matched the results from testing the complete hypothesis on pooled data, PMP's sample size and effect size. Both methods converge towards strong support for the true hypothesis. This means BS can reliably piece together evidence from partial studies to support the correct overall idea. Now, what about the interesting part when the hypothesis is wrongly specified? This is where things get a bit tricky. In condition 2, when they tested a misspecified hypothesis, the results started to diverge. This divergence was particularly noticeable when they tested the hypothesis against its complement. The complement hypothesis is basically everything except the specific hypothesis you are interested in. When there was no real effect, the results were similar to condition 1. But when there was an effect, but the hypothesis they were testing was wrong, things got weird, especially when testing against the complement. In scenario A, they saw that BS and the complete hypothesis test started to reach opposite conclusions when the hypothesis was misspecified and tested against the complement. In scenario B, this divergence was even clearer, as sample size and effect size increased. Best kept finding evidence for the misspecified hypothesis, while the complete hypothesis test correctly showed no evidence or evidence against it. Why does this happen? The researchers explain it using the idea of M-open and M-closed problems. An M-closed problem assumes the true model is among the ones you are testing. An M-open problem assumes the true model is not among the ones you are testing. When you test a complete hypothesis using pooled data, and the true hypothesis is one of those you are testing, it is an M-closed problem. But with BS, when you test a misspecified overarching hypothesis against its complement, it can become an M-open problem. This is because the complement itself might contain the true tested hypothesis. In their simulation, even though the overarching hypothesis was misspecified, some of the partial hypotheses were actually correct. So BESS, by combining these, ended up supporting the least incorrect overarching hypothesis when tested against the complement. This is a bit like saying, well, this isn't the perfect answer, but it is the best we've got among the options we considered. The researchers highlight that this divergence is not necessarily a flaw in B's, but rather a consequence of how it works and how we interpret it. It is not pooling data, it is synthesizing evidence from independent studies about how much each study supports a hypothesis. So, what is the big takeaway here? The authors emphasize that BS is a valuable tool for combining evidence from diverse studies, especially when traditional methods can't be used. It reliably supports correctly specified hypotheses. However, there is a crucial warning sign. They strongly recommend testing both the unconstrained hypothesis, which doesn't make specific predictions about order, and the complement hypothesis. If the unconstrained hypothesis shows no support for your idea, but the complement strongly favors it, that is a red flag that your main hypothesis might be wrong. This is because the power of testing against the complement is great when your hypothesis is correct, but can be misleading when it is not. This research is super important for scientists trying to build a coherent body of knowledge. It shows us that even with different types of studies, we can still get a unified answer, but we have to be really careful about what we're testing and what we're comparing it against. It is a reminder that the way we frame our questions, especially our alternative hypotheses, matters a lot. The researchers also suggest future work could look at how partial hypotheses that overlap more influence best results. This is definitely a fascinating area that will help us trust scientific findings even more. So, what do I think? This is really cool research that provides a practical solution for a real problem in science. The simulation study is thorough and the conclusions are clearly laid out. The main strength is showing how BS can handle heterogeneity in study designs. The biggest implication is the warning about hypothesis specification, especially the alternative hypothesis. It is a reminder that Bayesian methods, while powerful, require careful thought about the models being compared. The real-world implication is that researchers can now feel more confident using BS to synthesize evidence from their diverse studies, but they absolutely need to be mindful of checking their assumptions and comparing results across different alternative hypotheses. This adds another layer of rigor to evidence synthesis. That's it for today's dive into Bayesian evidence synthesis. What do you think about this method? Let us know in the comments below. If you enjoyed this deep dive, please hit that like button and subscribe to RUBIN'S Mind Lab for more science explained. We'll see you next time.